Happy Tuesday, and welcome to the notes that cover the second half of Chapter 21. Today's notes are about genetic drift and gene pool. First of all, genetic drift is just as the words imply here. It is the movement or the drifting of the genetic material. But before we do that, we have to make sure we understand what a gene pool is. A gene pool is all the alleles of a given population. If you look at Madison Grant, we have a limited gene pool here. If you go to Marian, Anderson, Indianapolis, they're going to have a larger gene pool because they have more diversity. There's two types of genetic drift that you need to be familiar with. The first one's called founder effect. And if you just look at the word, it kind of helps you to remember what founder effect is. Founder effect is like our founding fathers. So we had a population of Native Americans here in the United States, and then we had people come over from Europe and our founding fathers. And then our population here has changed. Our gene pool has changed significantly. So founder effect is a new introduced population that's different than the local population. The other type of genetic drift is called bottleneck effect. Bottleneck effect is where you have a severe loss in the population which causes a change in the gene pool of the population. For example, we have a fire, huge forest fire, and the animals that can run the fastest are the animals who get away, and they are the ones who survive, and our population from that point on tends to be the faster population. Again, bottleneck effect. So what are the effects of genetic drift? One, it can be significant in a small population. Again, you think of the Native Americans and our founding fathers. There weren't a lot of Native Americans, and it did make a rather significant difference in the population. It can cause rapid changes in the allele frequency. And we saw this when we did the lab the other day because, one generation would be almost all dominant alleles, and 2 generations later, it would be almost all recessive alleles. It can cause the loss of variation within a population. So again, if a certain population is not welcomed into a community, they tend to leave. And so we see a loss of variation. And then harmful alleles may become a permanent part of that population. Normally, in a large population, harmful alleles are going to kind of be filtered out. A smaller population, they definitely are not. And again, because of genetic drift, we will see harmful alleles becoming more permanent. All of this leads us to gene flow. Gene flow, again, just as the words say, is the flow of genes or the flow of alleles. And if we define it as the movement of alleles due to the movement of fertile individuals or gametes. Your book talks about how bees carry the pollen from one area to another area. That would be an example of movement. If you think of zoos today, they all have breeding programs and they're exchanging genetic material. And sometimes that's good because it helps to prevent inbreeding. But one thing it is doing is reducing it's going to be causing more common gene pools. So everybody's going to be related. And over a period of time, if we have something that affects that one population or that one gene pool, there is a good chance that they'll still yet go extinct. Yesterday in your notes, it was talking about what I call environmental selection as opposed to artificial and natural selection. Your book refers to it as adaptive evolution. And before we go into this, we have to understand what fitness is, or relative fitness, and those are used interchangeably. And we define it as the contribution an individual makes to the gene pool relative to other individuals. So how many of your alleles are going to be expressed in the next generation? If you're fit, there's going to be a lot of them. If you're not so fit, there won't be that many of them. We have 3 types of this adaptive evolution. The first one's called directional selection. So if you look at the graph, you can see our original population is the dotted line. And populations tend to be in bell curves if you graph the trait versus the number of organisms. So again, let's look at Madison Grant. We have short people, we have tall people, but most of us fall in that average height. So if you're graphing this, height is going to be on the y-axis and the number of organisms is going to be on the x-axis. So say we have a selection against an extreme. In this case, it's going to be the short people can't compete as well. Their legs are shorter, they can't travel as far, so they eventually starve out. What happens to our population curve is it shifts to the taller side. And again, you see this in this particular graph. So one extreme of the population is favored over the other. Population curve shifts towards the favored side. If you're talking Galapagos and you're talking the beaks of the finches, if you're on an island that has lots of nut trees, the finches with the larger beaks are going to be able to eat more. You're going to see more large beak. The small beaked finches can't eat those acorns, and so they're eventually going to starve out. Next one's called stabilizing selection. In this case, both extremes are not favored, and the average or the norm is favored. And as you can see from this graph here, the number of individuals there in the middle increased significantly, whereas either end, there were fewer individuals. And the third one is called disruptive selection. In this case, the average of the norm is disfavored, and therefore the population trends to the extremes of the bell curve. And in this case, an example of this one would be, okay, we've got a bunch of mice. We have a few white ones, we have a few black ones, and we have mostly brown ones. And maybe there is a volcano erupts and now everything is covered with ash. And so those brown mice are now very visible. The white mice learned a long time ago how to hide. The black mice are now hidden. And so you see the brown populations going down and the other two going up. In our lab the other day, we talked about heterozygote advantage. There was the last case that we did, the individuals that were heterozygous had an advantage over the either homozygous dominant or the homozygous recessive individuals. Classic example of this is sickle cell and sickle cell anemia and malaria. In this case, if you're homozygous dominant, you're susceptible to malaria. If you're homozygous recessive, you're susceptible to sickle cell. If you're heterozygous, you have an advantage over both. You have to know this relationship here, and I've attached a couple of videos here. The first one is long. It's a little over 14 minutes long. It gets into a lot of history. It's very interesting, and if you're bored, go for it. The second one is just a good overview. It's about 4.5 minutes long, but it kind of, again, delves more into this malaria sickle cell relationship. Okay, frequency dependent advantage. Again, just look at the name, frequency. How often is this going to be an advantage? And in this case, the fitness or the ability to pass on the alleles in the gene pool is dependent on the number of the individuals of the population with the trait. Your book talks about scale-eating fish and how some of them feed from the left side of their mouth, or the mouth is on the left side of their body, and some feed with their mouth on the right side of the body. And depending on how many you have of the same trait, depends on how much food you have. If you have a lot of fish who are left feeders, they're going to eventually start losing food sources, so the right side feeding population will increase. Or if you want to use another analogy, eating cake versus eating ice cream. If we have a population that survives on cake and we have a population that survives on ice cream, if you have a lot of people who are eating cake, eventually you're going to run out of cake, and so your numbers are going to start to dwindle, and the numbers of the ice cream eaters are going to increase. And then you start running out of ice cream, and then the ice cream population is going to start declining, whilst the cake population is going to start increasing. So what you see with frequency dependent advantage is that it's kind of a flip-flop. They keep flipping between one and then the other. Which leads us to sexual selection. How do we choose our mate? First of all, we need to understand that there's sexual dimorphism. That's the differences between the males and the females. Let's talk about, okay, when I think sexual morphism, I think of mallard ducks. And the females are that the natural tones, the browns and the cream colored, whereas the males are those beautiful, showy colored feathers. When we get into sexual selection, there's two types that you need to know. The first one's called intra. So this is going to be the same gender competing for the rights to mate. We talked earlier about white-tailed deer. Late summer, we have the bucks. First of all, they express dimorphism because the bucks have antlers and the does don't. But in the late summer, early fall, the bucks are going to start determining who is the number one buck. And they're going to push each other around, and they're going to chase the younger ones or the weaker ones off. And you're going to end up with your dominant buck, and he's the one who gets to mate with the herd. We also have what's called intersexual selection. And we see a lot of that this time of year. And in this type of selection, the mates choose their mates. So generally, the females are choosing the males, which is why when you go outside right now, you see male robins, and they have a really bright red, rosy breast, and they're singing up a storm and strutting around, trying to impress the females. And then the females make the decision on who she wants to mate with. So with all this evolution, you're asking yourself, why are there no perfect organisms? This has been going on for a long time. You'd think it'd be a perfect society, but there's several reasons on why. First of all, selections can only act on existing variations. We may think it'd be awesome to have X-Men powers, but unless we already have the trait for X-Men powers, we're not going to get it. Again, along with those lines, our selection is based on our DNA. Again, it's just what's accumulated over history is what we can choose from. We can't just pull something out of the air. Adaptations are compromises. You may be best in one environment, but you may not be the best in another environment. You might have thin feet, so you're going to survive really, really well on water, but you're not going to be as fast to run on land. And then lastly, chance, natural selection, and evolution are all interactive. They all work together. You may come up with something that you think is the ideal situation, and then all of a sudden the weather changes. Or you may be having the ideal senior year, and then all of a sudden COVID-19 hits and everything changes. So everything's interactive. Again, so all types of evolution and all things that affect how things do evolve over a period of time. This concludes our notes for today. Just a reminder, we have a test coming up. The test will be over 18, 19, 20, and 21. Really concentrate on 21. Make sure you know how to do those Hardy-Weinberg problems. There'll be some multiple choice. There'll be some short answer or essay type, and you should be able to get it all done within an hour. So anyway, that's coming up on Thursday, and I will talk to you later. If you have any questions, give me a buzz or drop me an email. I'll be listening.